The pancreas plays a vital role with regards to our endocrine system, particularly as it relates to how we process energy within our body. So the endocrine portion of the pancreas is comprised of collections of cells called islets of Langerhans. Now the pancreas will have both endocrine, which we'll talk about now, as well as exocrine functions, which we'll talk about when we go into the GI system. But the islets of Langerhans consist of these endocrine cells, two of which we will talk about. One are the beta cells, and these secrete insulin, and the others are alpha cells, and these will secrete glucagon. So before we get into talking about the actions of insulin and glucagon, let's review some terms that are similar sounding but very different. Glycolysis is the metabolism of glucose to pyruvate. Glycogenesis is the formation of glycogen. Glycogen is nothing more than glucose molecules bound to each other in a long strand. Glycogenesis is the breakdown of glycogen back into glucose. And then we have gluconeogenesis, which is the formation of glucose from non-carbohydrate precursors, such as amino acids, lactate, or glycerol. Insulin, our first hormone, is referred to as an anabolic hormone. And what that means is that it acts to store away fuel stores. It builds things up. It does so by facilitating movement of glucose into cells. It favors the formation of glycogen. That would be glycogenesis. It increases protein synthesis, and it increases the synthesis of fatty acids and triglycerides. So if we look at the actions of insulin regarding different food types, its actions regarding carbohydrates will be that it will facilitate the transport of glucose into skeletal muscle and adipose tissue. And it does so by facilitating the insertion of glucose transporters. The specific transporters here are referred to as GLUT4. Now, other tissues do not require glucose transporters, certainly, or at least not insulin-dependent glucose transporters. And those would be things such as neurons and erythrocytes, as well as some other tissues. Insulin stimulates the formation of glycogen in the liver. So again, glycogen is the storage form of glucose. It does this in the liver as well as skeletal muscle. Those are the two major sources or sites of storage of glycogen. Insulin will inhibit the creation of new glucose. So it inhibits gluconeogenesis in the liver. And it will also inhibit the breakdown of glycogen or glycogenolysis. With regards to protein, insulin will increase the transport of amino acids into cells. And in the process, then, it will enhance protein synthesis. It inhibits the breakdown of protein or proteolysis. And with regards to actions of insulin on the adipose tissue, it will, as mentioned, increase GLUT4 transporters on adipose tissue, and this will allow glucose into the adipose tissue, which will then be, through various biochemical pathways, converted into fatty acids and triglycerides. So adipocytes can take up fatty acids without insulin. However, lipogenesis, which is the creation of triglycerides, is something which is made much more efficient in adipose tissue by the presence of insulin. Stimuli for insulin secretion. Increase in plasma glucose will cause insulin to be secreted. Parasympathetic, which is our rest and digest system, stimulation of the parasympathetic system will result in increased insulin secretion. As we've mentioned before, oftentimes the autonomic nervous system has as a accelerator and a brake. In this case, sympathetic stimulation will inhibit insulin secretion. An increase in plasma protein levels will cause an increase in insulin secretion. And a substance called glucose insulinotropic peptide, or GIP, will cause insulin secretion. This will be a substance we'll talk about in the GI tract. Now, just as to give you a very rough idea here, what's the effect of insulin secretion, and that can be measured by plasma glucose. What's the effect of plasma glucose or insulin secretion to these different foodstuff? So if we eat a diet full of carbohydrates, we'll see that our plasma glucose and hence insulin secretion rises fairly rapidly and to fairly high levels. Protein, however, although it does cause the release of insulin, we'll see that that effect is much less, and then even less is the effect of fat on insulin secretion. But all will cause some degree of insulin secretion. Glucagon is essentially the opposite of insulin. It is secreted by the alpha cells, and its effects will generally oppose insulin. So whereas insulin was an anabolic hormone, glucagon is a catabolic hormone. So it tends to facilitate processes that will be breaking things down. The major sites of action of glucagon are on the liver and adipose tissue. So at the liver, glucagon will stimulate the breakdown of glycogen, or glycogenolysis. It stimulates the breakdown of protein, proteolysis. And realize that by breaking down protein and freeing up amino acids, that then acts as a substrate for the creation of new glucose through gluconeogenesis. It stimulates the enzymes for what we call ketone body formation. In adipose tissue, glucagon stimulates lipolysis, or the breakdown of triglycerides. So from this, it frees up fatty acids and glycerol and releases those into the bloodstream. The fatty acids can be utilized directly by other tissues through processes like beta oxidation to create ATP, or they may be transported to the liver, and there they can undergo an additional process, which is the formation of ketone bodies. Glycerol, which is also freed up at the adipose tissue, can be utilized by the liver for gluconeogenesis. Stimuli for glucagon secretion: a decrease in plasma glucose, sympathetic stimulation, increased plasma protein levels. And note that increased plasma protein levels is both a stimulus for glucagon and insulin. Now, let's tie together what we've learned about insulin and glucagon to understand the regulation of metabolism. So understand that as we eat throughout the day, our nutrient intake is intermittent, and yet the energy needs of our tissues are 24 hours a day. This means that we need to have efficient ways of storing this material, as well as ready access to these nutrients when we are not eating. An important underlying concept of this entire discussion will be the fact that our brain depends primarily on glucose for its energy needs. It can use ketone bodies, but glucose is its primary energy source. Now, as we talk about the regulation of metabolism, we think of two different states. One is called the absorptive state. So this is when nutrients are being absorbed from the GI tract. This is also referred to as the fed state. So during this time, glucose is plentiful, and it's the preferred source of all tissues. The post-absorptive state, or starvation state, is when our GI tract is empty. So in this case, we are having to mobilize endogenous storage of energy so that it can be used by our body. So during this time, we will see the breakdown of glycogen, freeing up glucose, glycogenolysis. We will see the creation of new glucose, gluconeogenesis, and we'll also see the breakdown of adipose tissue, lipolysis, and that will allow us to have ketone body formation as well as substrate formation for gluconeogenesis. The absorptive state is demonstrated here in this slide. So during the absorptive state, glucose is plentiful. All tissues will preferentially use glucose as their energy source. Our brain can uptake that glucose without the presence of insulin, as can the liver. It will uptake the glucose, and the liver, it will be used to form glycogen, glycogenesis. Now, during this absorptive state, insulin will be active, and insulin, its actions, we will see primarily will be affecting skeletal muscle and adipose tissue. So in order for these tissues to uptake glucose, we see that insulin will be necessary. So insulin inserts GLUT4 transporters into skeletal muscle and adipose tissue, allowing glycogenesis in skeletal muscle and lipogenesis in our adipose tissue. We'll also see protein synthesis can occur in skeletal muscle under the direction of insulin. Now, insulin will be inhibiting gluconeogenesis in the liver. It will also be inhibiting proteolysis. So all these actions that we see happening with insulin are all anabolic actions. We also have here that we will be inhibiting lipolysis. Now when we switch from the absorptive state to the post-absorptive state, we now are going to see that we have to free up this energy that we've previously stored away. And during this time, glucagon will be the primary hormone at work. So glucagon is going to stimulate glycogenolysis. This will free up glucose for use by the brain. Glucagon will stimulate lipolysis. This will result in the freeing up or the breakdown of triglycerides. Glycerol will be one of those byproducts. Glycerol can be taken to the liver and can be used for gluconeogenesis. Glucagon will promote proteolysis in skeletal muscle as well as in the liver, and in both of these cases the amino acids that are freed up can be used for gluconeogenesis. This formation of glucose will then be transported to the brain for use. The other byproduct of lipolysis, in addition to glycerol, are free fatty acids. The free fatty acids can be transported to the liver, and the liver has the necessary enzymes present to form ketone bodies. These ketone bodies can be used by other tissues for energy, as can the free fatty acids, and if necessary, ketone bodies can be used for the brain, or by the brain for energy. Glucagon is going to inhibit lipogenesis as well as protein synthesis. It also will inhibit glycogenesis during the post-absorptive state. So during the post-absorptive state, tissues other than the brain will primarily use free fatty acids or ketone bodies for energy. The brain will preferentially utilize glucose during this time, and if necessary, can use ketone bodies. But this entire process, all of these things that are going on, is referred to as glucose sparing. So we are sparing glucose for use by the brain, and we do so by having our other tissues use these other energy sources.